Hello everyone, myself Manju Sharma from Suresh Canvier University, Jagatpura, Jaipur. The department is School of Agriculture. Today we are going to discuss about diseases of onion and garlic. So the first disease is the basal rot, which goes by Fusarium oxysporum and Fusarium species, that is CP. These are the symptoms of the disease. The fungus produce many chlamydospores, which are thick-walled. Resting spores are microconidia, which are one-celled and thin-walled. Mode of spread and survival. The pathogen is soil-borne, and the optimum temperature for development are 28 to 32 degrees centigrade. Infection occurs through the root, either directly or through the wounds. Disease management. Basically, the growers must follow crop rotation and harvested bulbs must be thoroughly cured to reduce potential storage losses. Onions are very sensitive to low soil copper levels. In order to optimize crop production and disease susceptibility, additional soil copper fertility may be needed, especially on muddy and sandy soil. Soil drenching with copper oxychloride, that is 0.25%. The next disease is the downy mildew disease, which goes by Paranospora destructor, and these are the symptoms. White downy growth appears on the surface of the leaves. Finally, the infected leaves are dried up. Pathogen. The sporangiosperm are non-septate, long and swollen at the base. Sporangia are pyriform to fusiform, attached to the stigmata by their pointed end. These sporangia germinate by one or two germ tubes. The coinocytic mycelium is intercellular with filamentous hysteria. Oogonia are found in the intercellular spaces. Mode of spread and survival. The fungus attacks the seed stalks in a seed crop and has been found on and in the seed as mycelium, but true seeds do not help in carryover of the fungus from one session to the next. The main sources of pollination are the diseased bulbs used for propagating the crop in many areas and spores present in diseased crop residue. If infected bulbs are planted, the fungus grows up with the foliage produce sporangia, and these spread the disease to other plants. Then the disease cycle or dormant period. It is believed that the downy mildew fungus overwinter primarily as mycelium in infected onions that remain in onion fields or in nearby cultiles. The pathogen also can overwinter in perennial varieties of onion in home garden. It is suspected that spores of the fungus that persist in the soil may directly infect the roots of young onion plants. These plants become systematically infected and serve as focal points for infection in commercial onion fields. So they will be completely killed. These repeated cycles of spore formation can result in severe and continued epidemics of downy mildew if disease favorable environment conditions persist. Then the management, their management, like 3 spray with mancozeb 0.2% is effective. Spraying should be started 20 days after transplanting and repeated at 10 to 12 days interval. Then the next disease is the left blight disease or blast disease, which goes by the botrytis species, and these are the symptoms. Botrytis is the major disease of onion in cool climate areas. Light infections do not affect yield, but heavy infections causing major yield reduction can occur. Hundreds of white specks are seen on the foliage. The disease then spread very rapidly, and tops of the entire crop may be killed. So the disease then spread very rapidly, and tops of the entire crop may kill. Pathogen. Botrytis is characterized by its conidiophore, which present an appearance of grape bunch. The conidiophore are tall, erect, and branches are irregularly or dichotomously. So they are dark and separate. The terminal botrytis is characterized by its conidiophore, which present an appearance of grape bunch. The conidiophore are tall and present. So they are dark and separate. The terminal cells swell to produce sporangious temperature. So they are dark and separate. The terminal cells swell to produce sporangious ampullae. On each ampulla, numerous conidia arise simultaneously on short denticles. The conidia are high-aligned or tinted aseptate and globose to avoid. That is the disease cycle. So, how they will be spread? So, sclerotic on onion bulbs in cull piles, on other bulbs in seed, fields and on volunteer onion plants in commercial onion field produce conidia spores that infect leaves on sprouted bulb and onion plants in commercial field. Sclerotic, sclerotic on the surface of the soil that intact leaves on a sprouted bulb and onion plants in commercial field. So sclerotic on the surface of the soil in commercial onion fields also produce conidia that can infect the leaves of nearby onion plant. Sclerotia on leaf debris which produce conidia, ascospores like sexual spores that intact leaves. Because the ascospore are the result of sexual reproduction, they may serve as the source of new. The ability of sclerotic to germinate and produce conidia rapidly, like up to 4 times, the ability of sclerotic to germinate and produce conidia rapidly up to farmer soil. The sclerotic on the bulbs of volunteer onion produce conidia and that infect either leaves of the same plant or those onion plants in nursery. So the sclerotic on the bulb of volunteer onions produce contain in the absence of seed field and culp piles, it is suspended that sclerotic in the soil and sclerotic on volunteer plants have provide the primary source of inoculum for outbreak of botrytis leaf blight in commercial onion field. Because of ascospore are the result of sexual reproduction, they may serve as the source of new strain of the pathogen that are tolerant to fungicide used to control botrytis leaf blight. The ability of sclerotic to germinate and produce conidia repeatedly up to 4 times results in the production of conidia over an extended period of time. Sclerotic on the bulbs of volunteer onions produce conidia that infect either leaves of the same plant or those of onion plant in commercial fields. In the absence of seed field and kill piles, it is suspected that sclerotic in the soil and sclerotic on volunteer plants provide the primary source of inoculum for outbreaks of botrytis in commercial onion fields. Secondary spread, the dense, tangled growth of leaves that develop from bulbs in curl piles provided conditions, little air movement, and high relative humidity that are favorable for subsequent production of spore on dead leaf tissues. The results in secondary cycles of infection in the curl piles. Similarly, secondary cycles occur slightly later in seed field. Conidia are blown from the seed field and curl piles to commercial bulk production fields, and the disease cycle continue. Leaves of onion. Leaves of onion plants in commercial field can be infected by conidia that develop from sclerotic in the soil. These infected leaves also serve as secondary source of inoculum once conidia are produced on the dead leaves tissue. Leaves of volunteer onion plants infected by conidia that develop from sclerotic in the soil. These infected leaves also serve as secondary source of inoculum. Leaves of volunteer onion plants infected by conidia produced by sclerotic on the same plant also serve as a secondary source of inoculum. Leaves of onion plants grown for commercial production are repeatedly infected, and these serve as the source of inoculum for infecting new leaves throughout the growing season. So these infected leaves also serve as secondary source of inoculum once conidia are produced on the dead leaf tissue. Tier management, bulk treatment with Captain or Theorem 0.25%, spraying of MANAP or mancozeb or chlorothalonil. Fungicide may be applied every 5 to 7 days for disease control. Then the next disease is the Phytium root rot disease, which goes by the Phytium ephenidatum, Phytium debarianum, and Phytium ultimum. The symptoms of the disease are like this disease causes seed rotting, pre-emergence, damping off. The disease appears in circular patches in the field here and there. All the affected plants get killed. If the disease occurs prior to seed germination, it causes gappiness. The seed or seed material are killed before their establishment. The disease also occurs after establishment of the crop after 15 to 30 days of sowing or planting. This stage is called post-emergence damping off. If the disease occurs very late, it causes stunting of the plant and rotting of the roots. Then the pathogen is Phytium dermatum, their epidemiology. The fungus is mainly soil-borne. The disease is favored by ill-drained conditions in the field due to stagnation of water. High soil moisture or continuous rain may favor rapid development of the disease. Their management: seed treatment with TRM or Captain, that is 4 gram per kg. The bulbs may be dipped in TRM solution 0.25%. After sprouting, the root region of the plants along the rows should be given a soil drenching with copper oxychloride, that is 0.25%. Then the next disease is the Asthma disease, which goes by the Urositis schipuli, and these are the symptoms. Then the black Asthma disease. So the sori are seen at the base of the leaves and the leaf surface. Black powdery mass is seen after rupturing of sorus wall. Pathogen: the sori of Urositis schipuli contain dark colored and powdery spore masses. The spores are found in permanent balls. Each ball consists of an enveloping cortex of tin, sterile bladder like cells with one or two central dark colored, thick walled chlamydospores. So the spores germinate by means of short promycelium while still in the ball. Then the mode of spread and survival. The fungus remains viable for 15 years in infected soil by means of spore balls. It persists in soil as a saprophyte. Onion bulbs and onion transplants are important means of widespread distribution of the fungus. Implements also help in the spread. Wind-borne soil and surface drainage. Water are important means of local dissemination. Now their management: seed treatment with TRM or Captain 4 gram per kg. The bulbs may be dipped in the TRM solution, that is 0.25%. So these are the symptoms of black Asthma disease. The leaves become yellow and die back, and when the plants are pulled up, roots are found to be rotten, and the base of the bulb covered with a white or grey fungal growth. Later, numerous small black spherical sclerotia are produced, and the bulb of the onion completely rots. Then the mode of spread and survival. The disease is worst in warm summer, or in the case of winter, onion during warm spell in autumn or spring. Sclerotia persists in soil for 8 years. Then The primary inoculum consists of spherical small black sclerotia produced in infested fissures of allium species during previous years. Then the sclerotia are transported from field to field by flood water. Then the disease cycle. The sclerotia that form on the decaying host will lay dormant until a host plant's root exudates stimulate germination, especially root exudate that are unique to allium species. Cool weather is also needed for germination of sclerotia and hyphal growth. The soil moisture levels optimal for host root growth are also optimal for sclerotia germination. Mycelium will grow throughout the soil, and once it encounters a host root, the fungus will form aprocharia, a structure whose purpose is to aid in the attachment and penetration of the host. Now, their management, crop rotation and clean seed are the only effective control. Then the heavy manuring with organic manures reduces the disease in the crop. Then the seed dressing with benomyl, carbendazim or thiophanate methyl 100 to 150 kg gram per kg seed gives effective control. Now, the next disease is the purple blotch disease, which goes by the Alternaria pori. Symptoms are like this disease occurs mainly at the top of the leaves. The infection starts with whitish, minute dots on the leaves with irregular chlorotic areas on the tip portion of the leaves. Then circular to oblong, concentric, black velvety ring appear in the chlorotic area. So the lesions develop towards the base of the leaf. The spores join together and they spread quickly to the entire leaf area. The leaves gradually die from the tip downward. So basically, these are the symptoms of the disease, that is purple blotch disease. So the spots which will be purple in the color, that's why the name given purple blotch. Now their management, like disease free bulb should be selected for planting. Seed should be treated with Yaram 4 gram per kg seed. The field should be well drained. 3 foliar spraying with copper oxychloride. 3 foliar spraying with copper oxychloride 0.25% or chlorothalonil 0.2% or GNAP 0.2% or mancozeb also 0.2%. Now, the next disease is the neck rot disease, which goes by the botrytis alli, botrytis schistosa, and botrytis nerea. The symptoms are like usually appear after harvest, although infections originate in the field. The greatest epidermis infection originates in the field. Greatest epidemic development. Greatest epidemic development occurs when cool, 50 to 75 degrees Fahrenheit, most weather prevalent for greatest epidemic development occurs when cool 50 to 75 degrees Fahrenheit moist weather prevails for some days before or during harvest. If the weather remains dry, if the weather remains dry during harvest at the curling, losses found in storage are usually small, and then the symptoms are first seen as a softening of the tissue around the neck of the bulb, or more rarely than the bulb. Then their mode of spread and survival. Mode of spread and survival. The fungi that cause snake rot survive the winter or previously infected onion debris in the soil, in coal piles and refuse dumps, and in trash in storage shed. So these are the symptoms of the disease. Now, the Phytium root rot disease, which goes by the Phytium aphanidematum, Phytium debryanum, and Phytium ultimum. Symptoms are like this disease causes seed rotting, pre-emergence, dumping off. The disease appears in circular patches in the soil field, and there all the affected plants get killed. If the disease occurs prior to seed germination, it causes gapping as the seed or seed material are killed before their establishment. So the disease also occur after establishment of the crop, after 15 to 30 days of the sowing. This stage is called post-emergence damping off. If the disease occurs very late, it causes standing of the plant and rotting of the roots. Then the pathogen is Phytium aphanidematum, their epidemiology. The fungus is mainly soil-borne. The disease is favored by ill drained condition in the field due to stagnation in water, high soil moisture, or continuous rain may favor rapid development of the disease. Now, their management. Seed treatment with Theorem or Captain, that is 4 gram per kg. The bulbs may be dipped in the Theorem solution 0.25%, and after sprouting the root region of the fruit, the bulbs may be dipped in the Theorem solution, that is 0.25%. After sprouting in the seed region of the plants, along the root should be given a soil and drenching with copper oxychloride, that is 0.25%. And the next disease is the Asthma disease, which caused by the urositis schifuli, and these are the symptoms: black asthma disease. The sori are seen at the base of the leaves and leaf surface. Black powdery mass is seen after rupturing of sorus wall. Pathogen: the sori of urocystis. Black powdery mass is seen after rupturing of sorus wall, and the pathogen, the sori of urositis, chiefly contain dark colored and powdery spore masses. Each ball consists of an enveloping cortex of 10 sterile, bladder-like cells with one or two central dark-colored, thick-walled clay spores. So the spores germinate by means of short promycelium while still in the ball. Mode of spread and survival. The fungus remains viable for 15 years in infected soil by means of spore ball. It persists in soil as a saprophyte. The onion bulbs and onion transplants are important means of widespread distribution of the fungus, and implements also with download. Spread: wind-borne soil and surface drainage. Water are important means of local dissemination. So it persists in the soil as a saprophyte, and then the onion bulb and onion transplant are important means of widespread distribution of the fungus. Implement also help in the spread. Year management, like we have to apply the seed treatment with Theorem or Captain 4 gram per kg, and the bulb may be dipped in the Theorem solution, that is 0.25%. Thank you.